So without further ado, I'd like to introduce our chairman, Dr. Xinxin Ding. Dr. Xinxin Ding, also a Nanjing University alumni. So he graduated from Nanjing University with a bachelor degree. Then he came to United States to further pursue the Ph.D. Education, and got his Ph.D. from University of Michigan at Arbor. And then he actually joined the State University of New York at Albany in 1994 as assistant professor, and climbed all the rank to full professor. Dr. Ding actually joined the University of Arizona recently. It's not recent, right? It was 2017.
5 years already.
5 years. Wow, that's fast. I thought I should join yesterday. That's great. So Dr. Ding joined the University of Arizona as a department head in 2017, and actually he actually oversees this. Our department has been in a really very rapid expansion in the past 5 years. I have to say that department has been completely different from when I joined. That was 2009. Wow. Anyway, so Dr. Ding has published over 200 papers, mainly on the drug discovery, drug depositions, metabolism, and chemical toxicity. So I think he is very perfect chairman for our department, because our department is pharmacology and toxicology. He has his expertise in cyclone P50. That is metabolizing enzymes that actually metabolize all these drugs, small molecules, including chemicals and drugs. So it's really a very nicely balanced the two branches of the two, our true specialty in the department. In fact, in the past, we always we used to have a pharmacology centric or toxicology centric chairman. So Dr. Ding has been awarded numerous awards, and right now he holds over grants that worth probably over $1 million, with 2 R1s and P grants and other collaborative grants. So he's a very accomplished scientist. He's I think it's the APPET, right? The applied pharmacology, experimental toxicology.
No,
therapeutics.
You need to know that.
Aspect.
American Society of Pharmacology and Experimental Therapeutics.
Yeah, he is the elected chair, and I think he's soon going to be the chair for that. Anyway, so just before we hand the microphone to Dr. Ding, I just want to add 2 personal notes. Scientifically, I gained a tremendous help from him. You know, my main weakness is like I have too many interests. You know, it's very hard to concentrate. So when he arrives, he look at my portfolio and really suggests me to focus on several promising areas. Since then, I have developed a very strong program actually on fungal asthma, and recently on the COVID-19. So really, myself and a lot of colleagues are benefit from his scientific advice, and also for chairman, he's not one of the most responsive chairman I have ever experienced. I have 6 chairmen through my career. So anytime if you have any issues and comments and the concerns, you can email him or text him. He really responds right away and help us to solve all the things. So anyway, so today we have so why I'm saying that? Okay, why I'm saying that? So if you're looking for a job, then we actually have openings. So I actually help the department and the Xinxin to actually put advertisement. If you guys and actually Xiaobo is here. So you recently have I think Dr. Lemos, right, join us. So anyway, we still have openings. So if you are interested, look for those ads. Okay, so today Xinxin gonna sort of tell us about metabolic mechanisms, chemical toxicity in the lung. He gonna help us to go through his research many years of research on this area. So welcome, Xinxin.
Thank you for the nice introduction. And let me try to share this. Make that better.
So in the same department, right?
Yep.
Oh,
Yep.
Oh, nice.
We still have endowed chair position. Are you interested?
You were just always kidding.
All right, so so happy Chinese New Year is coming up. You can sort of remind me to wish everybody happy Chinese New Year. So I work on metabolism, and lung is one of my sort of focus organs. The if I can, okay, good. I don't know if people in the audience know much about the P450, so I'm going to assume that you don't really work on this system, but this is sort of I'm a P450 centric. I did my graduate thesis on this enzyme system. So the P450s, actually they're in both in microsomes, the ER membrane here, and in mitochondria. There are a few in there that's mostly responsible for steroid metabolism and vitamins and so on. So, but then my work mostly had to do with the microsome, which is the bulk of the enzyme system. So these are redox enzymes. So the red blocks here, proteins, heme proteins that are anchored in the ER membrane, and they require an enzyme called the P450 oxidoreductase, or also some people call it cyclone P450 reductase, CPR or PUR, to transfer electrons from NADPH to the P450, and then these are really powerful as a catalyst, powerful enzymes. They can do a lot of things that organic chemists cannot do in terms of the type of reactions they catalyze. But typically, they're considered a monooxygenase reaction. They would take one atom of oxygen from molecular oxygen and combine with the electron that was transferred over, 2 electrons transferred over from the reductase. They can insert one of the oxygens into just about any bonds and form typically hydroxylated metabolite, although you get all kinds of variations in there. So this enzyme has been studied for about close to 60 years now. So there's a lot has been known. I started my sort of graduate work by purifying these enzymes. My mentor's lab in Michigan was the first group that purified these enzymes and showed the world that there's more than one P450 enzyme. So now today, all the enzymes are known because the genome, human genome, mouse genome projects have been done a long time ago. So within the P450 gene superfamily in humans, these are human genes listed here. There are 57. So this is a slide I got from Fred Yangrich from Vanderbilt, who was a former postdoc in the Kuhn lab, as my mentor's lab as well. So the gene names were, like most other genes, were based on sequence identity among the different gene products. So they go from 1 to, I think in mammals goes to maybe 51 is the highest number. But then you can also classify them roughly based on the substrate and metabolize. So these first column here are the ones that primarily metabolize steroids, although some like CYP1B1 really does also metabolize foreign chemicals as well. But then, the numbers, some of these steroid hydroxylase numbers are based on the position of the hydroxylation on the steroid ring. So others are based on the sequence that they're discovered. I guess the bottom line is that there are many of these. So the xenobiotics metabolizing enzymes are this group that has probably most of the work done on, and because they metabolize drugs, just about 90% of the drug drugs on the market are metabolized by P450 enzymes. And you've probably heard about CYP3A4, which is the in human liver, the enzyme that handles about 50% of all the drugs that they can metabolize. Not necessarily the only enzyme metabolize a particular drug, but so there's a lot of different activities going on in academia, try to understand the genetic polymorphisms, the structure-function relationships, regulation at various different levels. And there's a huge activity going on in industry. The pharma really have to, the FDA requires them to screen all their candidates with these P450s to see if the drugs gets turned into something toxic, or if they're inhibited by known drugs and so on. So because that's the basis of drug-drug interactions or adverse effects and so on. And then you got some that seems to favor sort of fatty acids, the linear molecules, and so on. And then some are involved in vitamin. There's a bunch of overlap between the vitamin and the steroids for metabolist enzymes. And there's a few still sort of left as what Gingrich calls the orphan P450s. But these are the ones that have not had substrates like identified as definitively preferred substrates for them, although some like 2S1 have been found to metabolize cancer drugs like in a reductive, not in a typical oxidative fashion. So my lab, when I started to do my postdoc, have my own lab, I sort of wanted to do something that was different from my sort of graduate work. So I started to look at genetics and look at genetic approaches. At the time, the making a knockout mouse is still pretty sort of challenge. It's just beginning of the era of this approach. And so since then, we've made lots of different knockout mouse models to knock out different P450 genes, and made a few transgenic mice where we put the human CYP genes into the mouse, and then if you cross the transgenic to the knockout, then it's called CYP humanized. That means they're not all humanized, but just for that gene. But we also later on decided to take a sort of sledgehammer approach to knock out this reductase. Now, while there's many P450 genes, but there's really only one reductase in mammals. So if you knock out this reductase, then none of the P450s can be functional. So you can look at sort of collective functions, all P450 isoforms in different scenarios. We've also now, since sort of moving here, made a few more other related enzyme like the epoxyhydrolase, which is a non-P450 enzyme, but it's also sort of involved in metabolic mechanism. Heme oxygenase, I'm sure you're familiar with, but we have a project that's looking at the conditional knockout and then testing of this enzyme in a relationship with arsenic-induced toxicity. And we've also made a albumin humanized mouse model for biomarker related work. But the reason I'm showing you this is that if you have any interest in these models, then I'd be happy to share with you to do some interesting work. All right, so metabolism. So here's a substrate called NNK, which is one of the two sort of what's called tobacco-specific nitrosamines. This is the structure of the compound. They're only formed in tobacco product, and this is a well-known human sort of lung carcinogen. And the P450 enzymes metabolize it by attacking, this is the N-nitrosyl group. Compounds that have this functional group typically are nasty. They're many of the carcinogens have this. So the P450 catalyze something called alpha hydroxylation reaction. The alpha carbons next to the functional group here, this carbon and this carbon, are vulnerable. So they're metabolized in one or two pathways, and then eventually they form these diazonium ions that can readily react with DNA to form DNA adducts. So in the case of NNK, lots of studies have been done. A lot of this is done by Steve Hecht at Minnesota in the early stages. I think he's still working on this type of compounds now, but then he and his colleagues have shown that this 6-methylguanine is the main reason why these compounds cause cancer. And then, but they also form other adducts, and some of these compounds from this pathway here actually inhibits DNA repair. So the both sort of a 1-2 punch in these two pathways that really cause mutations. So NNK is a typical sort of genotoxic carcinogen, but this reaction really depends on bioactivation because this carcinogen, because this compound itself, if you don't have P450, the prediction would be that they wouldn't be carcinogenic. There's also sort of detoxification pathways, and this sort of ketone can be converted to hydroxyl group, and this NNAL would be substrate for conjugation enzymes, and which would make them sort of water soluble, more water soluble, and then excretable. So one of the sort of my favorite examples to show the ability of P450 enzyme to mediate carcinogenesis is this experiment. So we made this mouse model. It's a gene cluster knockout where we put a LOXP site at one. So okay, so backtrack a little bit. So these the focus of my group has sort of more on the CYP2 gene family within the P450 superfamily, and there within the P450 superfamily, there's a number of these gene clusters where the genes, through evolution, they duplicated and multiplied, and then there's gene conversion events happened. So there's a lot of genes with similar structure, maybe 95% identity, but then they have different substrate specificities and different regulations and so on. But this cluster of genes includes the CYP2A subfamily, 2B, 2F, 2G, 2S, 5 different subfamilies, all together in mouse. I think it's chromosome 7, and humans it's chromosome 19. And most of these are expressed in the respiratory tract, in the lung, the nasal mucosa, and so on. So have been biochemical studies done to show that these the enzymes in themselves can metabolize lung carcinogens. So we wanted to test the hypothesis that these actually, their metabolism does make a difference in the tumor genesis outcome. So we made this mouse model. I put the LOXP site at both ends, at the one at the 2F1, the 2F2 gene one and 2S1, and then managed to delete this. That was like 1.4 megabase or something like that. It was a quite feat to do this at the time. But with this model, we were able to show that this is a standard or typical lung tumor bioassay using NNK as the carcinogen. So just single dose IP injection at different, saline 0 milligrams per kg for NNK, and then 50, 200 mg per kg, just one injection, and you wait 4 months, then you have plenty of tumors to count in the high dose group in the wild type mice. And so if in this knockout, even at this high dose, you don't really see any tumors. So this very clearly demonstrated that these P450s are essential for NNK-mediated lung carcinogenesis. Now, to support this, the genotoxic mechanism, that we also lost the ability to form DNA adducts in the lung, this 6-methylguanine DNA adduct, that in wild type you would be able to detect quite a few abundant levels. So that was quite interesting. But then, when you think about it, these 100 mg, 200 mg per kg NNK is not a really environmentally relevant dose, and it's it's the nature of toxicology studies. You have to use high doses to be able to induce tumors as a model for study. But in reality, people are exposed to tobacco smoke. And in tobacco smoke, there are more than 4,000 chemicals and at least 200. This is kind of an old number. I'm sure a lot more now are known toxicants, carcinogens. So really, nobody knows which, in a sort of mixture exposure scenario, which carcinogen. You got the NCIs, and you probably have heard about the polycyclic aromatic hydrocarbons like the benzoyl pyrines and the aldehydes. Those all individually can cause cancer if you give a high dose in animals. So which one of those actually is important in tobacco smoke induced carcinogenesis? And of course, then, for our purpose, is P450 important in this, right? So bioactivation converts these chemicals, and not just NK, but benzoyl pyrene requires metabolism. A bunch of other chemicals in tobacco smoke requires metabolism to become toxic. The first thing that happens actually is inflammation, which eventually would lead to COPD and then cancer, perhaps. So we started to look at this more recently to try to look at the sort of a disease model based approach. So we first look at the lung inflammation. This is a sort of a 1 or 2 week tobacco exposure model where the mice would be exposed to by nose-only inhalation, 5 minutes per session with 5 minutes in between. So it's like four 5-minute sessions per day. And you do this daily for either 1 week or 2 weeks. And then at the end, and the controls would be exposed to filter air. So at the end of that time, then we measured, we did the lung lavage to look at cells, total cells, macrophage, neutrophils, lymphocytes, and also look at some cytokines, and also histology to look for signs of inflammation. So it's a complex slide here, but basically we have wild-type mice and this null, the CYP2 ABFGS null mouse that we looked at also for the NNK-induced lung carcinogenesis. So this, if we start from this left side, you get the control wild-type filter air, get some basal amount of cells, and then with ETS is what we call it's environmental tobacco smoke. Is a mixture of mainstream and side stream smoke exposure. It's kind of mimic what a secondhand smoker would be exposed to. So that smoke exposure was 1 week in the first 4 columns, or 1 week, second 4 columns are 2 week. And you would get a significant induction in total cells and other cells, macrophages, and so on, all of the same kind of results. And in the knockout, the basal cells, basal cell numbers about the same, but you still get a little bit of increase in cell numbers, but it's much attenuated. So this is suggesting that these P450s are important in tobacco smoke-induced lung inflammation. The response in 2 weeks is a little bit lower in this wild type group, and because by 2 weeks, I guess, and you guys know this more better than I do, that the response is not as acute. But then you get the same results here, that the knockout are less, basically non-responsive now, by 2 weeks. We did find some cytokines that have the same kind of trend, and then we find in over here in this histology slides that airways that are surrounded by macrophages in this ETS exposed wild type mice, but not in the knockout. In the knockout, we do see some macrophages, but they're more around the blood vessels, not in the airways in here. So bottom line is, yes, the P450 enzyme do have some roles in tobacco smoke-induced lung inflammation. So then we moved on to look at the tumor outcomes. These are more involved. The tobacco smoke, unlike NNK, where a single dose injection, you get like 30 tumors per lung. For tobacco smoke exposure, well, you do those typical protocol that's developed by Weechi's group in UC Davis. It has been used quite a lot. It starts, you start about 6 weeks time, you expose them to whole body exposure, and if typically 120 mg per cubic meter total suspended particulates level, and you expose for 5 months, and then you have to wait 4 more months to be able to see the tumors more clearly, because in fact, if you by 6 somehow the smoke exposure, if you continue to expose them, the tumors don't grow as fast. So, but in this protocol, by end of this 9 months exposure, then you sect the mice to do tumor counts and so on. You get about a couple of tumors per lung. So you require a lot of mice to be able to show a statistical significance. But anyway, so we have the wild type mice and the CYP2 ABF GS, this cluster knockout mouse, and the smoke exposure or filter air controls. And the results, this is the result from one of the first cohort that we did, where we have the wild type filter air control, get spontaneous tumor. Now these, you have to do it in AJ mice, which you probably know this is more sensitive to chemical induced lung cancers, and expose them to ETS. You get, in this case, 1.5 tumors per lung. So, but it's quite significant difference. And then in the knockout mouse, the N means knockout, you still get some increase, but it's not as great as in the wild type mice. So there's a significant difference between these the wild type ETS and knockout ETS exposed. So we've confirmed this in the second cohort of experiment of mice, and then the results are similar. So this data is exciting. This is the first time that someone has shown that P450 metabolism actually make a difference in tobacco smoke induced lung carcinogenesis. Now, of course, then, so the question is, people, whenever I talk about these kind of studies, people say, well, can we then inhibit or prevent carcinogenous smoke-induced, particularly smoke-induced carcinogenesis by inhibiting P450s, right? Yeah, I mean, according to this, sounds like it sounds like a good idea. But then this is mice, and humans are a little different. They have differences in the sort of smoke deposition. There's differences in P450 expression. The P450s are not 100% identical in sequence, so there will be need to look at human people. We've done sort of some work with. We have a humanized mouse model where we've expressed the sort of equivalent CYP2A enzymes in 2A, 2B, 2F enzymes in the lung, and we were sort of hoping to replicate this kind of experiment by looking at the humanized versus the knockout mouse model. So now to change a little bit, to talk about one of the chemicals that are also found in tobacco smoke, naphthalene, but it's more sort of recognized as it's not just the tobacco smoke, but it's a petroleum sort of in petroleum products, in all kind of in gasoline. It's the main compound in mothball. So it's one of the most, in fact, everybody in the world probably had exposed to this because it's a common pollutant. One of the highest abundance chemical, the metabolites are the highest abundance chemicals detected in the urine of humans. So this compound has been found, is well known to cause acute lung toxicity, airway toxicity, and has also been found by NIEHS, by the National Toxicology Program, to cause lung cancers in mice and nasal cancers in rats. So we started to do some more in-depth study using this compound as a sort of model compound to look at the acute toxicity, and also because this is a non-genotoxic carcinogen. It's up to this point, no one has shown that this compound is well, actually there's evidence now. We have the evidence that it causes DNA adduct formation, but the significance of DNA adduct formation in carcinogenesis has not been studied, but mostly because it's such an efficient enzyme in causing airway damage, and the damage is quickly repaired. So this repeated cycles, damage and repair, is believed actually to be the main reason why they're carcinogenic. But the OSHA, since people are exposed to this quite a bit, and like Air Force workers, for instance, are exposed, people, that's car exhaust and that. But so OSHA limit is 10 parts per million for this. So we did one study where we just wanted to ask the question of which organ is mainly responsible for naphthalene-induced lung toxicity. And we asked this because the liver is actually very active in metabolizing naphthalene, as well as the lung. And the naphthalene and other sort of volatile chemicals that industries, they like styrene, ethylbenzene. There are a lot of activity going on where industries try to sort of classify them as not as toxic as what researchers are reporting. And so one of the main arguments the industry is making is that, well, human lungs not as active as rodent lungs in the ability to metabolize a lot of these chemicals. So, because of the species difference, even though these chemicals cause cancer in rodent lungs, they're not necessarily carcinogenic in humans, right? But it's hard to sort of argue with that kind of argument, because human data, it's hard to come by. And you really, a lot of this is all projection, extrapolations and models and so on. But we thought mechanistically, we need to sort out, is the lung the only organ that can actually cause this bioactivation and cause the toxicity in the target organ, in this case, the lung, the airways. So this is one of the studies where we use this P450 reductase. Remember, there's only one reductase, so if you knock out reductase, none of the P450s can metabolize compounds like naphthalene. So this is a Krelok system where this is the reductase gene. Most of the coding regions were knocked out. We used a TETO-CRE system that we set from Cincinnati, generated many years ago, where it's a doxycycline inducible, specifically using, I think that it's using CCSP TETO-CRE system, so it's specific for club cells, and but that model also had some knockout in the type 2 cells, I think. So, but anyway, so this knockout, and then I won't show you the data on the effectiveness, but it caused deletion of reductase mostly in the airways, and I think about 80% of the airway epithelial cells. In the club cells, it's knocked out. So so we expose mice, the wild type versus the controls versus the lungs specific reductase knockout model in this nose-only exposure to naphthalene at 10 ppm, and then 2 days later. So that's enough to cause airway injuries. The 4-hour session is enough, and then 2 days later we injected BRDU, and then after 1 hour collected lungs to look for BRDU incorporation to detect the sort of proliferating cells in response to injury. And so this is an example in proximal and distal airways where this proliferating cells are detected. And the quantitative data is shown here on the right side. The filter air controls didn't have that much proliferating activity, which is expected. In the naphthalene exposed mice, the black bar is the control genotype. There's plenty of, it's about 20% of cells are undergoing proliferation in both proximal and distal airways, but in the knockout, lung-superior knockout, this extent is much reduced to less than 10%. So, all right, so this says, well, the lung local metabolism definitely is important to some extent, but it's not 100%. But this is not a perfect setup because the nature of this knockout is that we didn't get 100% knockout in all the airway cells. So it's hard to say whether this residual proliferation is or injury is because of the residual cells are getting damaged, or because that there's metabolites generated from other organs that come into the lung, as we hypothesize is causing this. So we did a sort of a complementary experiment where we knocked out this gene from the liver using the albumin driver. So this is actually quite complete. By 2 months, all the hepatocytes have lost the reductase. Now we tested this by the exposure, and then if you look at this toxicokinetic data, looking at naphthalene levels, the LCN, which means liver CPR knockout mouse model, they don't have reductase in the liver, so there's no activity, no metabolism, and the naphthalene level is higher, which makes sense because the liver metabolism is meant to help with disposition, with getting rid of naphthalene from the bloodstream, right? So if you don't have metabolism, then you get more in the circulation. Now, if you look at this data, you're going to predict, okay, these lungs going to be subjected to greater toxicity because you got much higher doses in the circulation. Well, when you actually look at the damages, and this is actually done in Laura Van Winkle's lab in UC Davis, and they specialize in look at the sort of morphometry to quantify the fraction of the airways that are damaged. But the green line, similar like the green line in here, is the knockout, and the blue columns are the wild type. And you can see that there's actually a difference, a lower extent of damage in the green, in the knockout than in the wild type. Now, this is opposite to what you would predict from the toxicokinetic data. So this actually is more conclusive evidence saying that the liver is producing some metabolites that gets to the lung to cause toxicity, because naphthalene itself cannot cause toxicity. So if you put this into a model, you got the airways, naphthalene inhalation exposure, they're absorbed into airway epithelial cells where there's plenty of P450 to metabolize it. The primary metabolite is this oxide. They can cause local toxicity. So that has been shown by this lung CPR knockout model. But naphthalene quickly gets into the blood and it gets circulated to the liver and other organs. In the liver, they can also be metabolized and produce naphthalene and a bunch of other metabolites. So based on this data, it's indicating that these metabolites can come out into the blood and go back to the lung where they could contribute to toxicity. Now, if this is true, then this whole sort of risk assessment model needs to be modified to not only consider the lung metabolic capacity in converting naphthalene to metabolite, reactive metabolite, but also the circulating levels of metabolites, or what metabolites should be considered, right? And I hope there's some chemists in the audience that would appreciate these structures here. So this is naphthalene, very simple polycyclic 2 rings, aromatic hydrocarbon. So P450s, and particularly the CYP2 cluster that we studied, they will form this epoxide, And this is a highly reactive compound that's got a half-life of less than a minute. It will react with thiol like glutathione, SUGS, glutathione transferase or not. These glutathione conjugates are considered to be less toxic. They're detoxification products. They're further metabolized into mercapturic acid and excreted into the urine, right? But there's another enzyme, microsomal epoxide hydrolase, or EPHX1. That's the one that we've made a conditional knockout for now. Is they'll convert this to a dihydroxylated compound, which is further converted to 1,2-ethylquinone. And you'll notice the red color here means a reactive compound. So this is a reactive, and then there's this 1,4, because this can also go down this way to form a 1,4-ethylquinone. So you got the 1,2 and 1,4-ethylquinones, which are reactive, but not as reactive as the 1,2-epoxide. And people have shown that if you inject 1,2-ethylquinone into mice, you cause lung toxicity. So clearly these are toxic, and if they're produced in sufficient amounts or circulating in sufficient high levels in the blood, when you're exposed to ethylene, they could be contributor to toxicity. But this one can also be further metabolized by P450 to form a sort of 3,4 epoxide, dihydral epoxide. This is analogous to benzoyl pyrene, which is the ultimate carcinogen. That compound is it's a diol epoxide. So this could potentially be also quite toxic, although not much has been done here. So the sort of the nightmare here for someone who studied metabolism of this compound is, okay, how do we know which one of these compounds actually, this 4, is actually responsible for DNA damages or cytotoxicities and cancer, right? So that's what we're trying to sort out now. So we have a knockout here. The goal is to see if you knock out this this enzyme, which has happened also in this pathway here down below, then you shouldn't have any of these quinones formed. So would that make a difference in lung toxicity or lung carcinogenesis, right? We're trying to sort out which P450 is doing this step here, and then that will also sort of give us some way to sort out this compound, whether versus this. And then we're looking for DNA adducts and protein adducts formed when mice exposed to ethylene, and that will also give us some insight into which one of these actually is reacting with macromolecules under in vivo conditions. So this sort of scheme, let me see what time it is now. Okay. This scheme is sort of is showing that a compound like tobacco smoke carcinogen ethylene is metabolized by P450 to reactive metabolites, and then you have detoxification pathways also. But then if you have an overabundance of the reactive metabolites, then these will overcome the sort of detoxification mechanisms, and then you can end up with DNA adducts, protein adducts, or other sort of cysteine conjugates. And then knowing what these adducts are, that will help us to sort of better predict risk of lung toxicity or lung cancer from exposures to these compounds. So that's what we're trying to do. And then I'm sort of talking too much today here, so I'll try to speed up a little bit here. So this is one of my students, Sarah Hannon, has made some progress in identifying what the DNA adducts are. I won't go into details, although Laura's lab in UC Davis, in collaboration with Lawrence Livermore, have used the C14 labeled ethylene to show that there's actually DNA adduct formation for sure, because using accelerator mass spectrometry, that's much more sensitive in detecting these adducts. We've also using this Albany humanized mouse model found that this naphthalene oxide, this initial epoxide, can react with albumin, human albumin, and we found that this adduct is much more stable than the glutathione conjugate, which disappears very quickly after it's formed following an exposure. So this albumin adduct offers I think a half-life we calculated is about 74 hours, which is decent for biomonitoring in humans. We're working with a guy called Jeff Burgess here, who worked with firefighters to see if we can detect this in firefighters and use this to gauge levels of exposure and bioactivation in vivo. I want to just spend quickly a couple of minutes to talk about this new sort of thing that Xinxin, of course, is knowledgeable about this because he's on this. One of my students' thesis committee, Lisa Tran, is working on this project. Is that one of the steroid metabolizing P450 is called CYP19A1, which is also the aromatase. So this is the enzyme responsible for converting androgens to estrogens. So if you knock out CYP19A1, then there's no estrogen production. So we sort of made a knockout, made a macrophage-specific knockout of this gene, and found that the mice cannot, following naphthalene exposure, the airway injuries are not repaired properly. They just not repaired. So we're trying to sort of look into this, see what we can learn about this model. And this is this experiment that was done. And you expose mice to naphthalene corn oil, wild type versus knockout, and look at the initial acute toxicity, which we didn't find a difference. And then 72 hours look at cell proliferation, where we saw a difference between the knockout and wild type. And then 14 days look at the recovery using club cell CCSP levels in the airways to gauge the level of repair. Then that's where we found that the knockout didn't repair well. So this knockout then is using lysine-Cre to drive the conditional knockout in these cells. Now, aromatase apparently is not expressed in none of these other monocyte sort of cells, but it's only in the macrophage. So that's why we call it a macrophage-specific knockout. So right now, this is an ongoing study, so I won't go into too much details. But right now, what we're looking at is that we have a phenotype. So this is a macrophage CYP19 is knocked out. And then we see a phenotype that is the epithelial repair following naphthalene injury is impaired, right? So we have some other observations. We see that estrogen levels is decreased in these cells. We see adrexin expression is upregulated. So adrexin is a protein that's really known to be involved in in the early development of the neural tube. But they do act through the Wnt signaling. So the idea is that they compete with Wnt ligands too. So if you have higher levels of adrexin, that would block the Wnt signaling, and that leads to changes in here that might explain why we have an impaired epithelial repair. So we're trying to do an adrexin knockout to sort of tease out this pathway. We're also wondering if this impaired epithelial repair would lead to greater carcinogenesis, and so we're hoping to do a pilot project to launch into that. So that's basically it as far as what we're doing with the lung. We're looking at some other organs as well. So this is people who contribute to the work in my group that has to do with the lung. Some people already left. Lisa Tran is one doing the CYP19 knockout. Sarah is one looking at the DNA adduct. Uyghur is also looking at CYP19. So whereas Xiaomeng here actually from Qinyu. So Qinyu is my wife, who's also a faculty member in the department here. So her lab has been working closely with us on these. Also, Xiaomeng's one looking at the protein adduct, and I have a long-term collaboration with Laura Van Winkle's group from UC Davis, and she's an expert on cell biology in the lung. And Buck Holt's group at Lawrence Livermore did the DNA adduct detection for C14 labels, and locally Jeff Burgess from Public Health. Minnesota, Rob Turisky is a DNA adduct, protein adduct specialist guy who worked together for a long time, and some other people. So just a few slides here to hoping that you would be able to visit us in Tucson, particularly this time of the year. It's very nice. Some images from my backyard, and this is from my window office, our sort of entry to the department here. I'm happy to answer some questions. I should have left a little bit more time, I guess. That's all I got. So thank you.